pretty quiet here.
Okay, great.
And I'll go down to this.
All right, well, let's start off with announcements as usual. Jeff, I'll give you a minute to stop chewing, but Jeff was just sharing some interesting news from the legislature, positive stuff. So I've been out on vacation for a week. I don't have a whole lot of announcements to share, but Jeff, what have you got for us?
So Clean Water Fund, of which Forever Green is funded between the $6 million. That is passed on the House floor. It's still in the Senate, but the bill matches in both House and Senate. So seemingly like good news. We're not at the finish line yet. But seeming like knock on wood that funding is come through. Thanks.
Thank you, Jeff.
Yeah,
Thanks, Jeff.
We're in a disaster.
Well, for those that don't know, the relationship around this legislative, these legislative items, when it comes to clean water legacy, it is in fact a true joint venture between University of Minnesota for Evergreen and the Department of Agriculture and the other agencies, right? So there's representatives from the agencies. Our main connection to clean water funds is through the Department of Agriculture. And without their support, those dollars would not be met, right? In terms of opening up the door for Forever Green to make the presentation of the ideas, right? So the agencies, especially the Department of Agriculture, plays a key role in putting these ideas, for example, from Forever Green in front of the water council. Obviously, so appreciate all of the hard work you guys do on behalf of Forever Green, the whole process, and it's a long, it's a long process. If you don't, those of you that don't know, it takes time, it takes effort on their behalf as well as our behalf to make sure that these ideas get heard in that legislative process.
The Starship Unified Labor Fund for plans of commercialization and marketing, I think so that part of the 6 million.
Oh,
That's a separate.
So that's several.
that would be part of the continuous living cover value chain development fund, or MDA has a different name for it now that I have trouble remembering.
Use living cover marketing initiative.
Okay,
And that program will be, in fact, managed through the Department of Agriculture, not the University of Minnesota. So those are separate items, but obviously connected.
yeah, so that, I think that's in the Senate bill, if I remember right. The Senate and the House are a little different between those two, and the Senate does earmark kind of the first $1 million, so to speak, for Kernza, elderberry, hazelnut, winter camelina, I think is what it is. I didn't know there was a start to be an article on that though.
So it was basically, there's agreement on the ag bill.
I just. Okay. Yeah, so that money would go to entrepreneurs, to folks that are building businesses, creating new products, being part of the supply chain, to complement to the research dollars that all needed to move the crops out there.
My daughter loved the puffs, by the way.
Really?
I didn't even need sweetener or anything. Just kept eating puffs, so.
That's awesome. Kid approved.
Yeah.
Any other announcements from online or in the room?
Well, we had representatives from the legislature on campus last Wednesday. We were supposed to give them a field tour, but the snow was still covering the fields. So we had the teams organize presentations at 11 sites in the greenhouse facilities. We had an hour and a half, so they were short presentations, but I think overall it went really well. And then they came back to 306 Borlaug Hall, and Beth Dooley served them a beautiful lunch that was well received. And the folks, our guests stayed until about 2:15, so they were here for a good period of time. And also were served Bang Brewery beer, so maybe that helped a lot.
I think we got in more crops than maybe ever before. Kernza, camelina, elderberry, hazelnut, barley. There might have been one more. So that all those different crops were used.
Alfalfa.
We should have.
Peas, oh yeah, we got most of them in. Well, that was the big debate, and Don always goes big. And it's not always right, but I wanted to make sure that this was an opportunity to show them the wide range of crops that are undeveloped. When we do it in the field, we don't have the ability to show them all, right? So this was a unique opportunity. So there's always downside to everything we do. I thought it went quite well. So.
All right, I think we're ready to go. Sajal, would you like to introduce yourself and then start your presentation?
Yeah. First of all, thank you to the Forever Green Initiative for inviting me to share this some progress on the work we are doing together with the Land Institute, USDA, K-State, and KAUST. So my name is Sajal Sthapit. I have a PhD in plant pathology from Washington State University, and I'm currently working with DeHaan at the Land Institute as a postdoc, working on developing an imputation pipeline for perennial grain breeding. So a broad outline of my presentation. So I'll be talking a bit about a challenge of perennial grain breeding. Then we'll talk about what we have done so far with genomic selection to address those challenges, and then finish with update on ongoing work on Skim sequencing for genotyping with imputation, what we have done on that line. So intermediate wheatgrass is the perennial grain crop that we are working on. It's been grown in the US for nearly 100 years now, mostly as a forage crop and for erosion control. That's what it was first introduced for. But what we are trying to do at the Land Institute is to domesticate it for an additional use of grain production for human consumption. In terms of genomic resources, we have a very good reference genome for intermediate wheatgrass, which is about 11.5 gigabase pairs, which helps us do all the genomic work better. So this figure, on the left, we see the root of intermediate wheatgrass of perennial, and on the right, the roots of annual wheat. So it kind of gives the rationale for why we are trying to domesticate this crop, which is the abundant amount of root biomass that it produces that persists in the soil through the seasons, which has the potential to greatly reduce soil erosion, reduce farm labor in terms of tilling and planting that's required every year for an annual crop. So if you are able to domesticate a perennial crop perennial crops like intermediate wheatgrass, it has huge potential for helping with not only food production, but also labor reduction and many ecosystem and environmental benefits. So with the annual crops, we have the benefit of domestication over thousands of years through the work of many millions of farmers over multiple generations. But the challenge with perennial grain that we are facing is how do we accelerate domestication of these mostly wild populations so that we can achieve the benefits we need in a short amount of time. Now, especially challenging with perennial grains is that we are faced with long selection cycles, long generation time. So the plant has to take several years before they can be phenotyped to select the next generation's parents. So that inherently makes the process slower. And we also found from Jared and Lee and other colleagues who work that the important domestication and agronomic traits in intermediate wheatgrass, they are controlled by many genes of small effects. So that makes genomic selection a good candidate where we can factor in the contribution of many genes to improve the population. Genetic gain over time or response to selection. So it's a very handy equation that guides our work. So it's essentially a product of selection intensity, like how many few plants are you selecting from a very large population? So that gives the selection intensity. Then selection accuracy, which is like the phenotype that we are measuring, how well does it represent our breeding goal? And then genetic variance. So we need to have a lot of variance for selection to act on. And the product of this is divided by how many years does it take to complete a cycle. And the main challenge with perennial grains is that like each cycle is multiple years. So that makes the genetic gain over time slower. So we are trying to reduce the denominator value through genomic selection so that we can make rapid progress. So like without genomic selection in perennial grain breeding with intermediate wheatgrass, so in spring of year one, we would cross the parents, and then in fall they'd be transplanted, the seedlings will be transplanted in the field. But then we'll have to wait till next summer to start taking phenotypic measurements, and only in fall of the year two we'll be able to select parents. And because this is a perennial, we also care about the yield in performance in years 3 and 4. So if you are to account for all of that, the breeding cycle can be multi-year, unlike in annual crops where you can complete it within less than a year. So with genomic selection, what we are essentially doing is we are looking at two subsets of population. There is a training population, which is phenotyped, as well as genotyped with a dense set of markers. And these two data sets are fed into to develop a genomic selection model, something that Jared has been leading, Jared Crain. So after feeding into a genomic selection model, what we are then able to do is just use genotyping data from the selection population to then estimate what the potential phenotype is going to be. So instead of having to wait 2 years to get information on phenotype, we can estimate what the phenotype or estimate what the breeding value would be just at the seedling stage. So then from the selection population, we take the genotyping data at the seedling stage in glasshouse, apply the genomic selection model, estimate the genomic estimated breeding values, and then based on that, we can select these seedlings for the next generation already, instead of having to wait for those 2 years. So through this process, we are able to speed up the breeding cycle time. But then as the population evolves, the genomic selection model also needs to evolve. So not only we take like the 100 best plants for crossing to make the next generation, we also pick about 1,000 of the non-best plants and then phenotype them also to keep improving the training population and keep advancing the genomic selection model building. So by applying genomic selection model, Jared and Lee have been able to advance, have the cycle time of intermediate wheatgrass, and with the ultimate goal of advancing 2 cycles per year, and in that way, like rapidly accelerate our breeding gains. So just some results from above, from multiple breeding cycles. So we see that from the genomic selection models that we have generated, there have been steady improvement in key yield traits like mass per spike, mass per seed, and then the free threshing trait. And also there has been steady reduction in seed shattering, so that we have more seed on the spike for harvesting. So the genomic selection work that has been done so far has been pretty promising, and we are getting close to 2 generations per year. And so genomic selection, it depends on having a large training population and having a large number of molecular markers that are genotyped. And then in this plot, like we saw that as the number of markers increased and the training population size also increased, the predictive ability also kept on improving. So there is still some scope to keep adding more markers and larger populations to improve our genomic selection model. And the work so far on genomic selection was done using genotyping by sequencing, GBS markers. We had around 18,000 GBS markers that we used for the model building. Now, GBS is a reduced representation genotyping system. It's based on 2 restriction enzymes. So the variants we get are like where those restriction enzyme sites are in the genome. So not all of the genome is represented, even though it can be very dense. And it does give us 60 to 70% missing data, but the data that we do get, like there is pretty high confidence on that. So this amount of missing data has to be imputed for GBS data to be used. But we are working on transitioning to a Skim sequencing approach, which is a whole genome sequencing or whole genome representation. So it's not, so it can get us variants that are like much more variants than GBS, because it's not limited by the restriction site presence. But the challenge is that at very low sequencing coverage, there can be like more than 95% missing data per sample. So it's much more missing data compared to GBS. Now, what do I mean by sequencing coverage? So sequencing coverage is just a number of reads multiplied by the length of each read divided by the length of the genome. So for intermediate wheatgrass with genome size of approximately 12 gigabase pairs. So if we sequence 12 gigabase pair per sample, then on average we have 1x coverage, which means each base pair will have at least 1 read, around 1 read on average. But we could like get 12 gigabit bases of read, but divide it up by 100 samples, by multiplexing 100 samples. Then each sample has a coverage of 0.01x. So what this does with gives us a lot of flexibility with Skim sequencing. We can generate a lot of data for a few samples and get high coverage, or we can do that for lots and lots of thousands of samples and get like low coverage data on the entire breeding population, which then after imputation can like go into genomic selection work. So what is imputation? So imputation is essentially a method of using the data that we have to fill in the missing data. And because in Skim sequencing, there is a lot of missing data. So in this gray panel, like you'll notice that the question mark represents the sites that have no genotype calls. So what an imputation algorithm essentially does is it has a reference panel which has like haplotype with genotype calls. And this can be provided by sequencing like parents or a diverse population at high coverage. And then the breeding population or the population of interest is genotyped at low coverage with lots of missing data. And then what the algorithm does is it uses the data that is present to figure out what segment of the ancestral haplotype it's inheriting, and then using that and then filling in the missing information from those haplotypes. So essentially by using very sparse data, you are able to get complete data sets, a near complete data set. And this has been used successfully in human genetics and animal breeding to get like a dense data set for genome-wide association studies and other gene discovery studies. Now, in our case, the imputation pipeline we used, what we did was we had 28 samples, 28 intermediate wheatgrass plants that were sequenced at very high coverage, around 10 to 30x coverage, so that the genotype calls made on these samples will have high confidence. So we use these 28 high coverage samples to ascertain variants, to find out where the variant sites are and what the reference and alternate alleles at those sites are. Then to test how well the imputation is working, we took the same 28 high coverage samples and then downsampled it to 0.02x coverage, which means we took the raw reads of these high coverage samples, and then we randomly sampled very few of the reads so that it would simulate what it would be like to sequence these samples at very, very low coverage. And then we use this algorithm called Stitch, described by Davies in Nature Genetics, which is designed for low coverage samples. So it benefits by having thousands or multiple thousands of samples genotyped at low coverage. So it's appropriate for a breeding program with large population. So we ran snitch imputation on 2,600 samples that were sequenced at low coverage, plus the 28 downsampled samples, which we can use later to compare how well the imputation is doing. And then after the imputation was run, so we compared the genotype calls made on the downsampled samples by imputation with the 28 high coverage samples without imputation to see what the match was. So in terms of variant ascertainment, in the 28 samples, we found lots of variants, which is a major advantage of using a whole genome sequencing approach like Skim sequencing. So on average, each chromosome had 3 to 6 million variants. So this is, I'm just showing the histogram of variant density on 1J chromosome and then 2J chromosome. So there are a lot of variants that we can work with, which kind of makes it, for genomic selection per se, we don't need this many variants, but having this information can be useful for like other kinds of studies down the line. So it gives us more flexibility. Just some results from chromosome 1J. So we ran imputation on like different density of markers because we want to find a sweet spot of like fast pipeline processing, but then also not compromise too much on the accuracy. Like running imputation on 3 to 4 million variants per chromosome, like it takes about half a day, but then because it requires multiple cores, you end up having to wait in queue for like several hours or days on the computing cluster. And then sometimes it also runs into error, then you have to repeat the run. So by like sampling smaller sets of variants, we can make the process go faster, but still have enough variance for our research needs. So on the right, we have a plot of concordance, which is the what proportion of the imputed genotype calls match the genotype calls made in the high coverage sample. And then the x-axis is info score. Info score is a metric that the stitch imputation algorithm generates. There is no like hard and fast rule. I mean, the general recommendation is to apply after imputation, apply a filter for info score of around 0.4 and select the variance above that for downstream analysis. But I mean, we find that above 0.2, like we are getting pretty high concordance of around 94 to 96%, and then kind of stabilizes after that. When we reduce the variance to around 180,000 per chromosome, I mean the concordance did not like change that much. It stayed around 95, and even at 46,000 variants, the concordance did not drop too much in the middle, but at high info score, the concordance dropped when we had like 46,000 markers. We also looked at concordance at different minor allele level frequencies from 0 to 0.05, 0.05 to 0.1, and so on to 0.5. Like it's all smooshed. So here also at like higher marker density, the concordance is kind of more stable across the minor allele frequency, with slightly lower concordance for very, very rare alleles. But as we went with fewer markers, it started to get a little wobbly, but still like stayed above 94% concordance. So it does slightly poor when you have fewer markers, but like it's not too bad in terms of concordance alone. However, concordance itself can be a little like misleading. For instance, imagine you have a marker that has like 95% major allele frequency and only 0.5% minor allele frequency. So for that kind of locus, by just guessing the major allele, you'll be right 95% of the time. So imputation quality score is another metric that like factors the like the percentage match just by chance based on the allele frequency. So like it corrects for allele frequency at a locus to calculate imputation quality score, and then the score ranges from -1 to +1. So negative value means that the imputation is actually doing worse than just imputing the major allele or chance, just chance. And positive value means that it's doing better than just following the allele frequency. So we see that imputation quality score is also pretty good after info score of 0.3 for this chromosome. But as we have sample fewer variants, then it kind of suffers at a very high info score. And at minor allele frequency, as expected, imputation score is not very good, but still positive at like very low minor allele frequency, but then it stabilizes at like a moderate minor allele frequencies. And in this case, when you had 46,000 variants, at the very extreme also, it kind of suffered a little, but still stayed in a positive range, showing that it's doing better than chance. So pattern on chromosome 2J was also pretty similar across. The imputation score and concordances were high or positive, but imputation quality score at minor allele frequency was a little more wobbly when we had 61,000 variants for this chromosome. So these are some of like our ongoing result from comparing the like effectiveness of imputation algorithm that we are using. Going forward, what we are working on is also taking the 18,000 GBS markers that we know the genomic prediction has served us well on, and then adding that to the variants that we discovered on the 28 high coverage samples, and then impute with those also to see like how well they do. And then we are also working on taking the GBS sequencing reads from the plants that we already have to see if we can use the GBS reads that have reduced representation and then combine it with reads from Skim sequencing to see if we can get variants called that were not like present in the GBS reads as well. So that's something we are looking at. And after all of that is done, in the end, like we'll generate different data sets that will be fed into developing genomic prediction models to see if Skim sequencing plus imputation is just as effective or better than what we have been using in the past 5-6 years. So that's the plan for the future. And I'd like to thank Jared from Kansas State, Steve Larson from USDA ARS in Utah, Jesse Poland, who is at KAUST, and Lee DeHaan at the Land Institute for helping with my work. And I'd like to take your comments or questions.
Well, thank you very much. So we'll open it up here for questions online. Can you see online?
Yep.
The questions online or within
In the chat box.
Hey, Sajal, can you hear me?
Yeah, yes, Lee.
Hey. Yeah, thank you, Sajal, for that summary of your work so far. It's been great. I just wanted to fill people in on how this work has came about. This is part of the FFAR funded work that is part of a grant that group at the University of Minnesota Intermediate Wheatgrass group led that. Probin and Jim led the submission of that. And developing this new marker system was a piece of that work, which was directed at enhancing the genomic selection in Kernza. And probably just a little bit more about what Sajal ended with there was the integration of the previous GBS work. So if we want to use our model that we developed over the last 5 years, we have to find a way to integrate the old marker system with the new one. Hence, we need to find this sort of imputation back and forth way to make the crossover from one marker system to the next. That's what Sajal's working on right now, and hopefully that's going to be successful. It's kind of the last piece to make sure that this is all going to work out for us. Part of the rationale for developing the system is, of course, getting more markers. It's the system that's probably the direction we're going towards as sequencing cost goes down. If we can continue to multiplex more and more samples, the sequencing cost will continue to drive down the cost of the genotyping. Also, GBS has been a proprietary system and always been a little bit clear about the IP around that system. So this is a method that isn't encumbered by any intellectual property problems. So it should be freely available to everyone if we get it working for wheatgrass.
Good to see you, Lee.
Good on.
Hi, Lee. I would just add in, you mentioned the FFAR grant led by Jim Anderson, but also the Kernza CAP has provided some of the
That's
funds
right.
well for a lot of the genotyping. Both those grants have been they're kind of looking at the same problem, but a little bit different method, but we're pooling data from both sources. So both have been really helpful.
Hey, Sajal, nice talk. Thanks for the update. So the first question I have is, when can other groups potentially try to implement, or at least, test out the methods, starting from sequencing all the way through imputation? Is it looking like a year, maybe further out than that? I just wanted to hear some thoughts.
Hey, Robin, I'll try to answer that.
Okay.
We've been asking ourselves that same question every cycle. So are we going to go for it this time? And so now, we're asking that question about our fall breeding cycle. So we will start those seedlings the 1st of August. We'll be genotyping them by the end of August, and so that we feel like we have to kind of have a no-go within a month or two here to buy the right supplies to go with SkimSeek versus GBS this year. But we're making a hard push, over the next month to really nail that down. And really that final piece is, can we impute across GBS and SkimSeek efficiently and effectively? So then there's all the questions about lots of little details and proving how well it works and whatnot. But I think we'll know pretty soon if it works for us. I think if we make the plunge and it works for us in August, everyone else can jump in soon after that.
Yeah, I was going to ask that. Like, are you taking 100% plunge towards the new technology, or are you going to do GBS as backup in case this kind of doesn't look too?
Yeah, that would be very expensive. I mean, that's one of the things we've thrown out there is we could GBS everything and SkimSeek everything, but it's just doubling the sequencing cost. We're talking, I don't know, $20,000 or more to do both at the same time. We have a lot of plants that we have SkimSeek and GBS on. I think it's 1,000, 2,000 Sajal that we have both of them on. So we can definitely test the methods against each other with that set. So hopefully if it works, looking backwards, we'll be good to go going forward. We have done a fair bit of SkimSeek, on plants from the past. So we know the method works well. We know we get good data. I don't think we have to worry about the lab, not being able to pull it off. It should be pretty straightforward in terms of the lab side delivering good data for us.
Is there an estimate on how much it costs per sample to genotype in this SkimSeek approach?
Yeah, so we've been hoping that the cost was coming down as the sequencing cost came up, but as that happened, the cost of plastic went up.
Right.
So the library prep has gone up at kind of the same rate that the sequencing cost has gone down. So we're looking at probably, Jared, maybe you help me out here, but something like including extraction kit, library supplies cost, and sequencing $6.
Pretty good.
Or $8 maybe. Maybe Jared can correct me.
I was going to say we're more like 8.
8.
They're getting the GBS and SkimSeek are probably per sample right now are pretty close, but the Skim sequencing uses the Illumina library prep that has TDE and TDE buffer, and I know one of the lab managers ordered some in January and it had went up 30% in 30 days or something. And the TDE's I don't know, 0.2 microliters and $7,500. So, but it goes for a long way.
I would just add that the technique itself in the lab, Praveen, is similar, if not easier than the GBS technique, so it's fewer enzymatic steps.
There's maybe one more normalization step.
Yeah, that's probably the biggest challenge is getting accurate normalizations, but I mean, to some extent, that's also required for GBS, so I'm not sure it's really is that different in that regard.
There are people looking at cost reduction methods, some sonication for fragmentation, and maybe cheaper ways of generating these libraries. So that might be coming down the line as well.
Well, looking forward to implementing it. It does look promising. I think there's some chromosomal differences that's, I suppose, to be expected. But on that note, Sajal, are you seeing more differences across other chromosomes? 1J looked really good. 2J was pretty good still. Is that the trend across the whole genome?
I think like others, most of the chromosomes look like 1J, but then there are like a few like that kind of look more chaotic, like 3J is all a little bit chaotic. But then we are also looking at different ways of sampling, like the variance, right? So after applying variance, after applying the filters, today I shared just variants that were randomly sampled, like to reduce like 10 times the density or 100 times the density. But then we are also looking at like how does it work when we sample, like we make uniform physical base pair bins and then sample within that, like how does that work? Or also in terms of genetic distance, so sample few near the centromere and then sample like more logarithmically as we go towards the edge, so that we can kind of simulate sampling across like genetic distance. So all of these, like we are going to see how that influences the distribution of variance, but also the imputation if accuracy, and then we'll be able to recommend, okay, what's a good way of reducing the marker density for this approach.
I have one last question, and I'll mute myself. If you're imputing, well, from scratch to getting imputed good data on 400,000 markers on, let's say, I don't know, 2,000, 4,000 plants in a breeding population, what's the computational resources you're looking at? How many days does it take, or what kind of memory and other things you're using?
Yeah, so for like 50 to 60,000, so the imputation has to run on one chromosome in a job, right? So it's like each linkage group has to have its own job. So right now for 50,000 to 60,000 markers per chromosome for like 2,600 samples. I've been running it on a 16-core multi-threaded total of 128 GB, and then it gets done in half an hour. If I go up to like 400,000, 500,000 variants, then it would take like 3 to 4 hours. And then for the like going up to a million, like 3 million variants. So when it's successful, it gets done between like 11 to 20 hours, depending on the chromosome. But then there is a high chance that it kind of runs into this memory allocation error. So then you'll have to rerun that chromosome again. So that's the kind of the trade-off, But for up to like 400,000, it's doable within 2 to 3 hours.
That's pretty good. I mean, that many markers is probably, like I said, already a bit of an overkill, but it's good to know. Thank you. Thank you for the update.
I have a quick question. In your simulations, how did you come to 0.02x? Or did you test other coverages?
So the cost is driving us. So we want to be able to make this as cheap as GBS. So if we have to do higher coverage than about 0.02x, it starts to get more expensive. So that's kind of our threshold cost, how much sequencing we can afford to do per plant. We've got to double that. It gets, it starts to get more expensive. The library cost doesn't go up, but the sequencing cost goes up. So if necessary, we could bump it up a little bit.
Yeah, it should be interesting in a simulation just to see what 0.03 or 0.04 does for accuracy. But yeah, I was just curious. Yeah.
I think Sajal did some of those other amounts, right?
Yeah, I mean, we tried like at 0.5x and 1.0x also downsampling, and it did not seem to make much of a difference in terms of like improvement in concordance or accuracy. And so like it, yeah, so that's why we stuck with the lower value. I mean, the stitch algorithm, it is kind of designed to work with low coverage. So having more coverage from one sample doesn't seem to do much, but having lots and lots of samples seems to help it better. So when we tried it at like 800 samples versus 1600, the 800 was worse than 1600 samples. And
Cool, good to know.
Steve Larson, do you want to ask your question that you put in the chat?
Sure, thank you. Sajal, what you might have mentioned this, but I had to step up for a minute during your seminar. What would be the requirements to implement this for other species?
Oh yeah, Jared can fill in if I miss something, but I think for SkimSeq, like other than the computing resources, the only thing we need is like good reference genome. Like we need a reference genome to be able to align and call, but like my understanding that with GBS in the early days, we were able to call variants even without a reference genome, right? So that might be the difference.
Yes, with GBS, I mean, early on, it was made to work without a reference. And with SkimSeq, I don't know that we necessarily need a gold standard genome, but I think you're going to want something that you're fairly confident is a representative, either contigs, large blocks, or something that looks like a genome. I would say, I mean, so you need some type of reference. On the lab side, the robotics to make the libraries is going to be highly important for normalization and micropipetting, 0.1 microliters or very small amounts. And then computationally, for a breeding program where we're going to genotype 4,000 lines, we got to have enough computing power nodes to be able to align all of those individuals to the reference genome. And then once we align it, then we can go in and impute. So I would say those are kind of the big things: computational power, the laboratory equipment, and at least a draft reference.
Jim, you got a statement earlier. I didn't catch it. What was it?
For my question.
Oh, yeah. Oh, okay.
Yeah, any other questions out there?
One more, sorry. How does that imputation pipeline handle heterozygous loci? Is it, I mean, it looks like you're getting, pretty good concordance, but I'm only curious on how it figures that out, especially on like an outcrossing highly heterozygous species.
I mean, it does give like heterozygote calls, but underlying, I mean, like it does. I mean, that's where the black box part of these algorithms come in. But what it seems to be doing is like, because it's low coverage, but we have thousands of samples. So for any site, it has information from like multiple samples for that one site. So like it generates internal like haplotypes based on that information, and then based on that it fills in, like makes the, first it produces the genotype probability and then dosages, and then if that reaches a certain threshold, then it's confident enough, then it calls the genotype. But that's the extent I understand, like how it's still a little bit of black box, as like any other imputation algorithm is to me.
I agree.
I think it's just got, it just has to make a probability of whether there's one or two haplotypes in that region. It looks at the data and says, you know, what's the most parsimonious explanation, given all the data across the whole population, is it more likely to have just one haplotype, or do you need two haplotypes to explain the data that you have, as sparse as it may be? I don't know if that helps, but
One of the issues we have with this genome is the amount of presence absence variation. So there's an inherent level of errors that are going to happen when we're requiring things to fit into the nice model, where at least one must be present, when in reality both of the haplotypes may be absent. Like there might not be anything actually there, and there's no way to handle that right now. Basically, you have to call it homozygous something, I guess, or call it missing. And so we end up with missing data that like can't be estimated. Then kind of what do we do with that?
Yeah, even in the high coverage samples, like there is about 20 to 25% of the sites like are missing, so they cannot find it even in high coverage.
Right. So you going from one plant to the next, you have 25% that's missing, and then about 25% that's new. So it's about 50% of the genome doesn't overlap between plants. So it's a kind of a thing that we haven't modeled well yet. I think no one's come to real grips with that. There's some of the things in maize where they have, you know, they've totally sequenced 100 individuals. You can start to do that, but when you only have one reference, that's all we have to go with.
I noticed that the focus is obviously on the grain, on the seed traits. And obviously that's key, right? But I noticed that here at the University of Minnesota, our breeder, the breeding team and ecosystem service team is starting to look below the ground. And I was, you know, you can, over time, you know, we show that root, I'm so damn proud of that root. And as we're increasing that grain component, you know, what is happening to the root. So what I'm wondering is, within the overall national program, is the University of Minnesota the only team that's starting to look at what's happening as you go through and make these selections, what's happening to the root system, or is that a common component of the breeding programs across in Canada and across the US?
Yeah, don't mess with our roots.
We're relying on you guys to figure it out.
Well, that's very supportive.
Yeah. I have a little project that I'm doing on seedlings, looking at the root and shoot growth of seedlings, just to start with, is the easiest thing. If you look at a wheatgrass seedling, the root to shoot ratio is much smaller than a wheat seedling. So that might surprise people. That you think that this rooty plant would make a lot more roots relative to leaves when it's young, but it's actually it's kind of a strange perennial grass in that when it germinates, the shoot seems to grow a lot faster than the root does. And I think this might be at the root of some of our problems with establishment. We learned about this from actually a forage agronomist, I think out of Iowa, first mentioned that wheatgrass was notorious for poor establishment due to slow root growth. So that the germinating seeds, the shoot starts to come out, but the root doesn't get into the moisture fast enough. And at least around here, we get a little bit of moisture, and then the next day it might be late August, but it might be 95 degrees and windy the next day, and the whole soil dries out before it comes up. So I'm interested in selecting for increased rate of seedling root development. So I don't know if it's going to go anywhere yet, but I'm just looking at the heritability of that and whether it's going to be possible to put that into our genomic selection model as a way to hopefully improve establishment. I don't know what that's going to do in terms of mature root shoot ratio. Obviously, if it's got kind of a low root development as a seedling in the species, but overall, you know, after a year old, it's got 4 times or 5 times the root biomass of a wheat plant. Things change when it gets older. In terms of how these above ground traits relate to below ground, we're very anxious to see your results on that.
Yes,
Yeah, I was just gonna, yeah, I think it's, I mean, this is some of the work that I think Ravina is we're starting to think about too, is not just the growth below ground, but the architecture, you know, like what if in a world you selected for a high yielding variety that had a architecture that wasn't as conducive to nitrate uptake, for example. I don't think that's gonna happen, but the traits of the roots, as much as just the sheer growth, I think is an interesting point.
and the other issue is that as you increase the weight of the seed, is anyone paying attention to stem strength in the context of lodging?
Yeah.
I mean, I've been working on that for 6 years, maybe. The somewhat frustrating thing was that it doesn't appear to be stem strength at all. So it never lodges based on the stems breaking. It's always what you might call root lodging. So it's at the base of the crown, be able to hold the plant up well. And I've always wanted to do some dissection of crowns of plants that lodge versus stand well. And I think you'd find that there's a lot more adventitious root growth really holding that plant upright for the plants that don't lodge than the ones that do. The lodging often seems to be, if you have a single plant, I don't know if you guys have seen this, but if you walk out of these single space plants that are 3 years old and you get a windstorm, you'll find that all the stems in the middle are lodged. They're like twisted up and laying down, and then everything around the outside is standing upright. So they're like little islands of lodging inside the standing stems. I've looked at height. Lodging and height are usually negatively correlated. So the tallest plants lodge the least, which is another frustrating thing. So stem strength had no correlation to lodging, and height, the taller they are, the less they lodge. That's not helpful because we know that physics, it's not physics driving that, it's something else. So it's genetic correlations about
Norman Borlaug is really upset.
Yeah, so it's like other things in the population. So it's how well rooted those tillers are is helping to drive the yield and make them, you know, a high yielding tall plant, tall stem, and
so Lee,
hold it up.
have you considered looking at the effect of strigolactone on those seedlings? Because it will give you a enhanced taproot, less lateral roots, and possibly a more vigorous response. If it works, then the methods for selecting at the seedling level for high strigolactone production are well established in the rice literature. And it would give you a breeding target, because that what you described is classic low strigolactone production.
First time I've heard of it. That's interesting.
I can send you a, let's see if I'll send you a review.
Yeah, definitely.
In the chat.
That sounds very interesting.
There you go. But it's just a, you know, it's just a shot out of the dark, but that's a typical phenotype. You get low strigolactone, high strigolactone increases taproot and also increases adventitious rooting, but suppresses lateral roots.
Yeah, so I've grown a number of wheat and wheatgrass seeds in some of these blue papers where you can see the development, and definitely wheat. You've got kind of the primary seminal root, but it also sends out about 3 or 4 additional roots about the same time.
So I don't want you to get the impression that I have all this information cataloged. I was writing a review yesterday on on that topic, and so you just sent it off to my co-authors to look at. So I actually have it up here, but it's not going to stay very long. But I think that might be, at least because people have looked at that, and there's actually some literature in other grains of looking at that might be, I hate to bring biochemistry to plant geneticists, but sometimes it's helpful. And you can buy a analog of strigalactone. It's called GR24. You want to write that down? From like Phytotech sells it. Strigalactones themselves are pretty hard to get, but GR24 is a analog that works, or an agonist that works quite well. Okay.
Yeah, thanks.
Any other items from anyone? That was a great, great contribution. Great idea to consider. Any comments, Lee, from your group? Any other items you want to bring up?
We'll keep you posted.
So the big Kearns intermediate wheatgrass meeting is going to be in the Twin Cities. Is that correct?
That is correct. Twin Cities, and the dates are June 22nd to 23rd. Should be a registration email coming out, I think, early next week. The Kearns a cap all hands meeting is the day before, Wednesday, the 21st of June. So we'll have a few days in the cities. The meetings and field tours, some imbibing of various Kearns products, and grad student poster session sounds like it's going to be happening. So good opportunities for grad students to share their work. Yeah, it's going to be great.
Well, thanks to all of you that presented today and contributed today. We appreciate your team and setting up the presentations. It was great update, and look forward to hope seeing everyone here in the Twin Cities in June.
Thanks.
Thank you.
Sajal.
Yeah, take care, everybody. Bye-bye.
Have a good weekend.
Yeah, you too.